Hi, hello, welcome to a Physionic detailed study analysis. Today we're going to be going over the effect that oxalates have on causing cancer. So we're going to go over a study. We're actually going to go over two studies, but really the 95% of the work is going to be focused on one study, looking specifically at the effects that oxalate exposure has on causing cancer. So we're going to go over some mechanisms, we're going to go over some cell work, we're going to go over some mouse work, and then we're going to go over a little bit of human research as well. So if you're not familiar with who I am, my name is Nicholas Verhoeven. I'm a PhD candidate in molecular medicine, and this is what I do. I break down studies so that you can understand them in a far easier format. So either you can agree with my conclusions or you can disagree with my conclusions. And if you disagree, please feel free to comment and critique so that maybe I got something wrong. And then I can pin an amendment to this topic, which will, of course, have your name as well as a thank you for making science better. All right, with that introduction out of the way, let's go ahead and jump into this. All right. So I'm going to give you a background on oxalates. Then we're going to go into oxalates and their direct effects on cancer. And then we're going to go over long-term exposure to oxalates and cancer growth. So really, if you get exposed to oxalates over time, what happens to cancer growth? And then specifically hone in on breast cancer and oxalate levels, which we have some human data for that. And then for my insiders, if you're part of my insiders program, you also get access to oxalates pro-cancerous mechanisms. So actually going subcellular and understanding how oxalates, if it turns out that they have an effect on cancer, how they have that effect, and then different effects of oxalates on different cancers, which is really fascinating information, which is actually going to ultimately educate us on our final conclusion as well. So if you're interested in accessing this section for the insiders as well, then just hop on over to my Physionic Insiders, and you'll get access to this as well as a whole host of different other videos, full unabridged videos with summaries and a bunch of other good stuff. There's tons of stuff in there. I'm not going to go into it right now, but there's a lot there, so you're always welcome to join. With that said, let's go ahead and jump into a bit of background on oxalates. Okay, so oxalates are really simple. I just have one slide on them. Oxalates are just a compound. For anybody that's interested in the chemistry of what it looks like, it's a really simple structure, a lot of oxygens associated with it. It doesn't really matter about the chemical structure, but I like to show it just as so people understand that what molecules look like. So this is the chemical structure. We're going to be using this as our representation. And oxalates are found predominantly in plant-based foods. So there's sometimes some tiny amounts in meat, but the vast majority is found in plant-based foods. So with that, let's go on and actually look at the data. And that data comes from this study right here. Oxalate induces breast cancer. So pretty inflammatory title, as short as it is. So this is study 195, and it is publicly funded. If you'd like to know more information on the funding source, as well as more information on the study itself, the DOI number and all that good stuff, which is actually listed right here. But I have that all in my notes that are attached to this video for your convenience. With that, let's go ahead and jump into the first piece of data, which is looking at the data on the direct exposure of oxalates on a variety of different cancer cells. So what we're looking at here is the researchers have literally taken the cells, so a number of different cell lines, and just plated them onto a plate. We do this in the lab all the time. And then what they want to know is if these cells are exposed to oxalates, is there an effect on cell growth or cell division in general, proliferation? And the way they're doing this is measuring the DNA level. So if you think of a cell has a nucleus, and that nucleus contains DNA, or the genes that then allow that cell to operate, or at least long term. And if you have one cell with a set amount of DNA, well, when it splits and it makes copies of that DNA and copies of everything within that cell and makes two cells, then the DNA levels will then increase. The total DNA in the dish will then increase. So that's what we find here. So on the vertical axis, we see the amount of DNA, which is a proxy for cell division or cell growth, really cell division specifically. And so the more DNA you have, the idea is, and it's not a perfect representation if I'm completely honest, but the idea is that then you have more cells in general. And we're looking at MCF7 cells, which are breast cancer cells, MDA MB231 cells, which are breast cancer cells, MCF10 cells, which are breast tissue cells that are non-cancerous. They are non-cancerous, to be clear. And then HEC293 cells, which are cancer cells coming from the kidney specifically. So what the heck does this show? Okay, I'll put that aside. Let's just ignore that. Okay, so now we're looking at the different conditions. So the different conditions are the control, which is where the cells are just applied. They're regular media, nothing else has been manipulated. This is the media, the liquid that would keep the cells alive. It's filled with nutrients that the cells need to survive. Then FBS 10%. So FBS is actually a very common addition to cell media. It would also be added to the control most likely. However, the FBS here, what they don't show, or what they probably should have said instead of FBS, is that it's FBS plus EGF. So EGF is a growth factor, which is specifically known to increase the proliferation or the division of cells in general. So this is what's known as a positive control. The researchers know what is supposed to happen, and they're adding it so that they can detect the sensitivity essentially of their model, of their experiment. And then OX 20 micromolar and OX 50 micromolar, those are the oxalate conditions. So additions, you don't need to know what micromolar are, just know that it's an increased concentration with a bigger number. 20 is lower than 50, 50 is higher than 20, so therefore it's a higher level of oxalates that are added to the dish. And then acetic acid, 20 and 50 micromolar as well. So acetic acid is added just in case. So in these other conditions, they're not actually pipetting anything into the actual solution. So with the acetic acid condition, they want to know if they pipette anything into the dish, is there an effect? So is it an effect of oxalates, or is it an effect of just pipetting something into the dish? And the addition to this is that it also changes the pH of the dish. So they're curious if the change in pH would have some sort of an effect. So what we're most interested in is the orange and the red. So these bar graphs right here, or these bars right here, at 20 and 50. So 50 being the red and 20 being the orangish color. Okay, so now that we understand all that, we can read this pretty easily. So the control, which is the blue, again, no manipulations have been added, it's just the cells, and we're comparing everything against the control. And we see that there's a statistical increase in the green condition, which is the FBS plus EGF. So there's supposed to be an increase in this condition because the condition is specifically designed to lead to more cell division and cell growth as a whole. So we see that that's true for the MCF7 cells. We see it's true for all the conditions. However, for the oxalates, we also see an increase. So this would indicate that the exposure to oxalates would then increase the number of these breast cancer cells. And the same is true for the MDA MB231 cells. And then more oxalates leads to a greater effect, or we technically can't say that because they don't actually compare between the two, statistically speaking. But my visual, I would say that there's probably at least a slight increase in the number of cells with greater oxalate exposure. Finally, the acetic acid conditions, there's nothing that happens in either one of these breast cancer conditions. What's fascinating, however, is that also the MCF10 cells see an increase with the EGF. However, there is also a teeny tiny increase with the oxalate conditions. It's really hard to detect, but they do see a slight increase in the number of non-cancerous breast tissue or cells. And then what's super fascinating is in the HEC293 cells, which are again the kidney cancer cells, there's no increase with the oxalate exposure or the acetic acid exposure. And there's the predicted increase with the FBS plus EGF. So that's really fascinating because it would mean that there's more specificity towards the breast cancer cells. Now we're going to go into more detail on the difference in that specificity. If you're part of my Insiders program, that's one of the areas that I'm going to be hitting on. So looking at some of the mechanisms and explanations for why. Okay, so that's this data. However, what we haven't done is then the researchers then hone in on these MCF10 cells and then expose these cells for a longer duration of time. So this is done over 3 days, so constant exposure over 3 days. And these are exposed over 7 days, so for a whole week, so a much longer exposure, especially when you're talking about cells. And this is specifically all in the cells that are non-cancerous, so the breast tissue cells that are non-cancerous. And we see that there's different concentrations of the oxalates, but in general, with oxalate exposure, we do see an increase in the number of cells, or the proliferation or the greater amount of these cells. Now, in addition to that, what should be noted, however, is that the vertical axis is quite different. So here we're looking at less than 2,000, and at the peak, we're saying maybe 9,000, which is still below this level at 10,000. So is this huge sudden proliferation of these cells? The answer is no. It is compared to the control, but it is not compared to cancer cells. So there is growth, but it's only at the highest levels for long exposure, and that exposure only leads to really small changes compared to the cancer cells or the EGF at higher concentrations, because here we're using 1%, here they're using 10%. So that's really important to note because it could lead to improper conclusions. All right, the next thing that they wanted to look at was to look at mice, and these mice are known as BALB/c nude mice. And you might be wondering why they're so ugly. Well, I don't know the answer to that. But I agree with you if you were thinking that. Maybe I'm just projecting my own thoughts, that these mice are quite, unattractive. They're kind of like that cat, which, excuse me if you have one of these cats, the cats that are literally look like nude cats. I mean, maybe they are. They're hairless cats. Yeah, it's sort of similar to that. I'm sure there's a lot of differences, but they look very similar. So they're using BALB/c nude mice, and that's actually going to be important later on. So I'm going to be adding some nuance to why that's especially important later on. So all they're doing in this situation is that they're exposing these mice, injecting oxalates into the tissue of these mice, and specifically into the breast tissue of these mice. And they do that over time. And we're going to see a graphic in the next slide that actually explains exactly how many times they did it, and over what length of time. And then what they found was, as a spoiler, what they found is that these mice ended up growing tumors. So they ended up growing cancerous tumors. And here they're measuring the size of the tumor. So here we have the comparison to non-cancerous versus the actual tumor size, which is what we're most interested in. And we see that there's a huge increase in the actual size of these tumors. So that's what we see imaged right here. So with continuous exposure to oxalates, we see tumor formation. So now to give a timetable and to give more context to this, here's where we can see these survival curves. So here we've got the number of days that the mice are alive. So 0, obviously at birth, and then they go all the way up to about 90 days. And the survival at 100%, if all the lines are at 100%, then that means that all the mice are alive. And as they drop down, then you see fewer and fewer mice that are still alive. And in the green line represents oxalate exposure to the breast. Then they've got a control exposure, so an injection of saline to the breast, and then they've got acetic acid injection to the breast. And they're doing this exactly the same for all the 3 different conditions of mice and seeing what happens. And the injection happens at all these time points. So across, it usually happened a few times a week. And they were injecting, if I remember correctly, 810 micromolar amount of oxalates, of saline, or of acetic acid. And with that, then you see an overall, the injection over time. Then the pink arrow, which is a little difficult to see, but the pink arrow is the indication of the first tumor that was detected. Okay, so these red are the actual injections. The pink is the tumor, the first tumor to show on the first mouse. And then the purple is the first tumor to show on all the mice in that condition, which turns out to be specifically the oxalate condition. So purple is different from pink in that all the mice now have at least one tumor, and some of them may have multiple tumors. And what we find is that if we look at the oxalate injection in the breast, that we do see at a certain time, let's say 80 days or 79 days, something like that, we start to see that mice start to die. And they die pretty quickly. So within 10 or so days, definitely within 10 days, all the mice have then died because of this tumor, or most likely because of this tumor that starts to appear in the oxalate injected mice. However, the other injections did not cause any tumors, and all the mice ended up surviving. So this, and you can see that the timetable also changes. So, 0 to 30, and then it continues, and then they don't actually see a change until day 60 and beyond. And then the other mice end up living to 160 and 200 days old. So they continue to live full lives. Now, that's one piece of data. Now, the other piece of data, which I found absolutely fascinating, was this piece of data right here. So it's the exact same conditions. However, now they're adding oxalate exposure to the breast, so same as what we saw over here, but now they also have a control to the breast, which is also found here, plus oxalate to the back. So they're injecting it in a different site, so not the breast, but the back, and then they've got a control in the back. And what they find is that when oxalate exposure, these are the same indications, when oxalates are exposed to the breast, we see the same effect. However, when it's exposed to the back, there is no death. Oxalates have no effect on, we don't see any tumors, we see no cancer, et cetera, et cetera. So that was really, really fascinating to me. The next step is to then look at the amount of oxalates that are found in the tissue. So the actual tumor tissue, are oxalates actually found in that tissue? So not just from injection, but do we actually see it kind of aggregating in that tissue? And the answer is yes. So if we look at non-cancerous tissue like we did earlier, or if we look at tumor tissue, we see that the amount of oxalates per milligram of tissue, because these is really small amounts of tissue, there's a significant increase in the number of oxalates per milligram of tissue. So just to create that relationship between oxalates and the tumor itself. And here's where we can actually look into some of the human data as well, because the human data, which is displayed here, is what they did is they took breast tissue from humans, from cancerous breast tissue compared to non-cancerous breast tissue, and they also found the same relationship, that with the cancerous tissue you see higher levels of, I should say specifically breast cancer tissue, we see higher levels of oxalates that are present in those tumors, and we see very low levels in non-cancerous tissue. Hey, if you're interested in having greater access to me and be able to join in on live sessions and be able to ask me questions, as well as have access to my full library of content that's completely unabridged, that also comes with summaries, as well as having access to an insider podcast that pulls together all the content that I release for the month, as well as many other benefits and an insider community, then I strongly recommend that you join the Physionic Insiders, which is my bread and butter, full-fledged premium service. So if you're interested, then hop on over to the links. And with that, let's jump back to the content. Now, I'd like to pull in this other review to look at some of the context, because we've looked at animal studies, we've looked at some cell studies, we've looked at some very intriguing data. However, we should contextualize this in terms of how it affects us on a day-to-day basis. So I'm going to do a little bit of that in this video, and then I'll probably end up releasing more on oxalates in the near future. So I hope to do that and add even more context to what I'm going to be providing here. But for the time being, there are a few points that are especially important. So this is study 196, and it is they received no funding for this, and there are no conflicts of interest. Okay, so let's apply some nuance here. So this review specifically looks at human data, and there isn't a whole lot of human data that's out there. However, what human data is available does give us some information. And they also don't have to necessarily look at human data. They can also just look at the amount of oxalates that are found in foods, absorption, things of that nature. And then we can glean some information from that. So what do we actually glean? Well, one is that we generate oxalates. I generate oxalates. I am toxic. Just kidding. But I do generate oxalates, and so do you. So one of the byproducts of our metabolism through a variety of different reactions within our cells end up generating oxalates, and then our kidneys end up excreting those oxalates. And it turns out that most of the oxalates that we're exposed to on a daily basis are actually generated from our body. So that's one aspect to seriously consider. Now, that doesn't mean that if you don't consume oxalates that you don't absorb oxalates and therefore increase your burden of oxalates. That is true. However, still the majority of those oxalates that are produced are still coming from your own cells. The other thing is that oxalates come in different forms. There are soluble oxalates and insoluble oxalates. We only absorb certain types of oxalates. So we only absorb soluble oxalates. And those are the most destructive in that they can chelate and bind to different minerals and things of that nature. And I'll be getting into more in that in a later discussion, but not for this video. But the point is that oxalates are not actually absorbed that well. So if you consume a total amount of oxalates of, let's say, 1 gram of oxalates, so 1,000 milligrams of oxalates, the actual absorption level is going to be dependent on the split. So if you have 800 milligrams of non-soluble oxalates, you're not going to be absorbing that. So it's a moot point. It's just going to go through your gastrointestinal system, and you're just going to poop it out. Done. Now, what actually gets absorbed is soluble, and then hopefully your kidneys will be able to then excrete it, which is what it typically does. And there are experiments that show that, yes, when we consume oxalates, we end up excreting it. That said, that doesn't mean that it doesn't necessarily have an impact before it gets excreted, but the amount of oxalates that we consume are quite low. Even highest consuming levels of oxalates are still quite low. So we're talking about maybe in the thousands of milligrams, and that's really high consumption of oxalates. The other thing is that if you're cooking your food, oxalate levels get reduced dramatically. I mean, I'm talking massive reductions. We're talking anywhere from like 40 to 90% elimination of oxalates just by heating your food. If that's by steaming, if that's by boiling, etc. Again, these are all things I'm going to be discussing in future content as well. So you would have to consume mass amounts of these cooked foods to ultimately then split that based off of soluble and insoluble, and then for the soluble to then be absorbed into your system. So there's a lot of different steps where the oxalate burden gets decreased substantially. The other thing is, we also now know that, or at least we have some idea, I should say, because this is mouse work, we have some idea that if you get exposed to oxalates in general, that may not be as bad as when the breast tissue specifically gets assaulted with oxalates. So could this still be a worry? Possibly, but this nuance, this context is incredibly important. Now, one area where it might be a little bit more of an issue is if a person is consuming raw, specifically plants. So they're consuming large amounts of just raw foods over and over and over for long periods of time. Could that be an issue? Honestly, I think that we need a lot more data on the topic to be completely clear on the specific effect that oxalates have. That is not to speak to plants. Plants have many other nutrients that are beneficial to our body. I know carnivore enthusiasts and whatnot may not want to hear that, but there's a lot of data. I'll be going over a lot more of that data in the future. However, plants themselves usually lead to better health outcomes. So the discussion here is limited to specifically oxalates alone. So do oxalates have an effect? Well, let's look at this midpoint conclusion. All right, so the midpoint conclusion at this point is that site-specific oxalate exposure induces breast cancer in mice with immune deficiency. This is the other context that I really wanted to apply here, is that the BALB/c nude mice are immunocompromised in a particular way, in that they don't have a thymus, or they have a non-functioning thymus. Now, they repeated these experiments in BALB/c mice that were not nude, which means that they're a different strain, a different iteration of the BALB/c mice, which do have a functioning immune system. And guess what happened? Nothing. They did the exact same experiments and nothing happened. They did not develop tumors. So this is a hyper-specific situation. And this is why adding this context, adding this nuance is incredibly important, because with all that information taken into consideration, my worry level decreases substantially. We're not just talking about a mouse model, we're talking about a very specific mouse model, and you use other mouse models and suddenly all these effects are eliminated. Now that still doesn't eliminate the problem that we see with the human breast tissue cancer cells that were exposed to oxalates, but that also is cell studies, and not discounting them, but the exposure to oxalates may be much higher than what you would actually see physiologically. So, however, oxalates do not cause cancer in non-immune deficient mice. So that's my point that I just made. Nor do oxalates cause other forms of cancer. So it was, again, specific to breast cancer. And if you had this influx of the exact same amount of oxalates to other areas of the body, again, if we were to specifically translate animal research to humans, which is always difficult to do, then if we just put that aside for a second and we just apply oxalates to any other part of our body, we do not see the production of these tumor cells. This is hyper-specific. Additionally, consumed and absorbed levels of oxalates, which I mentioned earlier, are significantly different. So you can consume X amount of oxalates, but the amount that's actually absorbed is much, much lower. With absorbed levels being lower, like I said, much more data is needed to make conclusive statements about oxalates. My worry level is low, but it's not non-existent. And I would really like to see a lot more data on all these different areas. And when you look at, for example, research, again, this is associative research, but when you look at associative research in humans, looking at the highest oxalate consuming vegetables, like let's say soybeans are one of the highest levels of oxalates that you can consume, the breast cancer levels tend to be reduced. Now, there's a lot of confounding variables in there. I completely acknowledge that. But if oxalates had such a profound effect at causing breast cancer, you would see a stronger association, not a decrease in breast cancer, but at least a slight increase, even if the association is kind of moderate, but we see an association in the exact opposite direction. If it's causative, why would that be the case? So maybe because other confounding factors prevent the effect of oxalates. That's a possibility, and I'll be going into one of those factors in the future, in future content. So as it stands, we need much more data, but I still wanted to cover this study because I've heard a lot of people talk about oxalates and their effect on cancer. Now, if you're part of the insiders and you do want to know more about the actual mechanisms and how it affects different cancers differently, then certainly you're welcome to join the insiders program, and we'll get more into that. But as it stands, that's where we are. Hopefully you feel a little more educated. At least you understand the study. I hope that you understood the study. And again, that context really matters. So it was a pleasure speaking with you, and I'll hope to speak with you in the near future. Bye.